Assalamu alaikum everyone, welcome back to my channel. This is part 2 of vitamin B12 deficiency. So in today's lecture we are going to study how vitamin B12 acts as a cofactor for enzyme. Vitamin B12 acts as a cofactor for two enzymes, methionine synthase and methylmalonyl-CoA mutase. First of all, we will study how vitamin B12 acts as a cofactor for methionine synthase. So methionine synthase is an enzyme and it catalyzes the conversion of homocysteine to methionine. And then this methionine is required for further production of S-adenosylmethionine, also written as SAM. So this methionine synthase requires a cofactor, that is vitamin B12. So a cofactor is a non-protein chemical compound or a metallic ion which is required for an enzyme's activity as a catalyst. Or we can say that the cofactor is a helper molecule of enzyme. Enzyme can work properly with its cofactor. So vitamin B12 will help methionine synthase to convert homocysteine to methionine and then to methionine to S-adenosylmethionine. This is the overall reaction which shows how homocysteine is converted into methionine. Okay. So first of all we have 5-methyl tetrahydrofolate. This means that with tetrahydrofolate, it is methylated as per position. Okay, so the 5-methyl tetrahydrofolate will give off its methyl group to vitamin B12. So vitamin B12 will take the methyl group from 5-methyl tetrahydrofolate and will be converted into methylated B12. This methylated B12 will then transfer its methyl group to homocysteine so that with the help of enzyme methionine synthase, homocysteine can be converted into methionine. And then this methionine is further required for production of S-adenosylmethionine. S-adenosylmethionine, also known as SAM, will then methylate DNA as well as produce polyamines. So as I have told you that the methionine synthase required vitamin B12 as a cofactor. Okay. So on the other hand, once this 5-methyl tetrahydrofolate is demethylated, once it removes its methyl group, it will be converted into tetrahydrofolate, which is active form of folate. Okay. Then this tetrahydrofolate, the active form of folate, will be then required for production of DNA. It will produce nucleic acid. So when there is deficiency of vitamin B12 in the body, if the vitamin B12 is deficient in the body, its level decreases in the body, which means that defective methionine synthase will be produced. Okay, so methionine synthase will fail to convert homocysteine into methionine. So once it fails to convert homocysteine into methionine, then level of homocysteine in the body increases, whereas level of methionine in the body decreases. Okay, as methionine synthase also take this methyl tetrahydrofolate as a substrate and indirectly regulate the level of tetrahydrofolate. So once this methionine synthase is defective, then 5-methyl tetrahydrofolate will not be enough methylated. So it will not be methylated, so the level of 5-methyl tetrahydrofolate will increases. So once 5-methyl tetrahydrofolate accumulates in the body, which means that this tetrahydrofolate level decreases. Okay, which means that there will be decreased or impaired production of DNA, production of nucleic acid, due to which enlarged erythrocytes are formed. Okay, enlarged erythrocyte are formed, which leads to megaloblastic anemia. Okay, and if the level of methionine decreases due to deficiency of vitamin B12, then it will lead to neurological disorder or neuropathy. Okay, the increased level of homocysteine will lead to endothelial damage and will lead to cardiovascular diseases. Okay, So this is how homocysteine is converted into methionine. This is another diagram which shows that this is methyl tetrahydrofolate. It will be demethylated. It will give its methyl group to vitamin B12 and it will be converted into tetrahydrofolate. Then this vitamin B12 will catalyze the conversion of homocysteine to methionine by an enzyme, that is methionine synthase. As this methionine is required for cell proliferation, protein synthesis, RNA synthesis, and translation, the methionine will then further produce S-adenosylmethionine, which will produce polyamines. It will also produce S-adenosyl homocysteine. S-adenosylmethionine will undergo methylation of DNA, RNA, phospholipids, and proteins. Okay? And it will be again converted into homocysteine. So you can pause the video for a second and you can read the slides. So in vitamin B12 deficiency, methionine synthase fails to convert homocysteine to methionine. As a result, pyrimidine bases cannot form, thus slowing down the process of DNA synthesis, which further leads to megaloblastic anemia. The other enzyme is L-methylmalonyl-CoA mutase, whose cofactor is vitamin B12. So, the essential step in lipid and protein metabolism is degradation of propionate. Okay. During the degradation of propionate, L-methylmalonyl-CoA is converted into succinyl-CoA by L-methylmalonyl-CoA mutase enzyme, which requires vitamin B12 as a cofactor. So this is propionate. Okay. So propionate will be degraded, it will be converted into acetate. So during its degradation, methylmalonyl-CoA will be converted into succinyl-CoA with the help of enzyme, that is L-methylmalonyl-CoA mutase. This enzyme requires vitamin B12 as a cofactor. Okay. So this vitamin B12, when there is a deficiency of this vitamin B12, then it will fail to convert methylmalonyl-CoA to succinyl-CoA, which means that the level of methylmalonyl-CoA in our body will increase. So level of methylmalonyl-CoA in our body will increase, whereas the level of succinyl-CoA in our body will decrease, because the enzyme for conversion of methylmalonyl-CoA to succinyl-CoA is deficient, okay, is dysfunctional. So the accumulation of methylmalonyl-CoA and the accumulation of its precursor propionyl-CoA and the odd chain fatty acid So these odd chain fatty acid, which it will not be metabolized completely. Okay. So this propionyl CoA, due to its accumulation, it will be replaced by acetyl CoA. So this acetyl CoA will undergo demyelination in nerve cells, which ultimately leads to neurological manifestation. So this is methylmalonyl-CoA and it is converted into succinyl-CoA with the help of enzyme methylmalonyl-CoA mutase, whereas vitamin B12 acts as a cofactor. So if vitamin B12 is deficient, the level of methylmalonyl-CoA increases, the level of succinyl-CoA decreases, which will ultimately lead to neurological defects.